All right, extracellular matrix. Um, so we talk about cell adhesion and paracrine factors and juxtacrine factors, and this doesn't happen like in a vacuum, right? Which bears lead on my next slide. That's probably where I got that term from. So we'll talk about the stuff that's in between cells, and this is extracellular matrix. For this image, this is a Picasso-inspired extracellular matrix, so that's why it's looks like this. I'm going to eventually start running out of artists, so I might have to Google them. MC Escher would be fun. A lot of Renaissance painters. Andy Warhol. Maybe we'll do that one next. Anyways, let's go. So next we have, so like I said, cell-to-cell interactions don't happen in a vacuum. I'm going to get my pointer out quick. So they occur in accordance with environmental conditions surrounding the cell. So this is the extracellular matrix, which is this network of macromolecules submitted by the cell. In plants, which I work a lot with, it's like lignin and things like that that are glycans and things like that. These macromolecules form a region of non-cellular matrix in the interstices between the cells. Cell adhesion, migration, and epithelial sheet formation all depend on cell forming attachment with these matrices. So they're made up of these in humans' cases, made up of collagen, proteoglycans, and specialized glycoproteins such as fibronectin and laminin. So remember that when we talked a bit about the diffusion of paracrine factors and signaling like that, they can travel along these extracellular matrices with the glycoproteins and the proteoglycans. So remember that, like, if they're in one conformation, they can be inhibitive, and if they're kind of built like a tunnel, then they can facilitate the distance that these things travel. Fibronectin is a large glycoprotein, which is an adhesive molecule linking cells to one another and to other substrates, such as bone, which is in a cell, but right, so we have like cartilage attaches to bone. Fibronectin has multiple binding sites, and interactions with the proper binding partners results in the proper orientation of the cells within this ECM. And as we said before, fibronectin also lines the roads over which cells migrate, so they can be also used for that to kind of, when we get to neurulation, we'll talk about how glial cells climb along as well. So they're very important, right? So extracellular matrices are made up of collagen and proteoglycans, as we said. This stuff, my notes I've read to you already. So laminin is a type 4 collagen, which makes up the basal lamina, which is a layer. As you can see, we have the epithelium, which is the tissue, and we have this basal lamina, and then collagen on top of it. So this is part of the extracellular matrix here. Basal lamina form these closely knit sheets. So you can see it's kind of a sheet. It's not really fibrous, just a flat sheet that underlie this epithelial tissue. And adhesion of these epithelial cells to the laminin is much greater than the affinity of mesenchymal cells to fibronectin. And this requires release for them to migrate. We'll get into the migration stuff when we get into the gastrulation and organogenesis stuff. So this is kind of an example here of an extracellular matrix. We have integrins, which are receptors for extracellular matrices molecules. So these are integrins. So they integrate the extracellular matrix with the cell itself by binding these proteins. So their job really is to connect the cell through this transmembrane domain here. They can have intercellular domains too, but the extracellular domain interacts with, in this case, this is fibronectin, or it could be any other proteoglycan or anything like that. Let's see. The main fibronectin receptor, which we show here is the screen, was found to be an extremely large protein. As you can see, it's quite big. I mean, this isn't to scale because it's an image, but they've depicted it quite big. and it binds fibronectin, spans the membrane of the cell, and attaches to the actin cytoskeleton inside here, right? So it links the interior scaffolding with the exterior scaffolding, right? So actin is this, microfilament that, organelles move across and proteins can be transplanted across. It's kind of like the highway system, right, within inside the cell. And so these integrins integrate or connect the extracellular matrix to the intercellular scaffolding. They're called integrins because they integrate the inter and extracellular scaffolds. I just get ahead of myself. I should just read my notes a little bit more. In addition, they also signal from outside the cell to inside the cell, causing changes in gene expression. So when we will eventually talk a little bit about the juxtacrine factors, which is in the next segment, but they can integrate with or communicate via other ligands on other cells or extracellular matrix. So this information is still able to be perceived by these integrins and passed and change gene expression. Remember, that's usually through conformation changes and then phosphorylation on the inside of these cellular walls. So let's look at kind of an illustration of how integrins relay signals from the extracellular matrices. So mouse mammary gland tissue divides when placed on tissue culture plastic. So if there's no basal lamina, this mammary gland tissue divides. The gene encoding cell division, or these proteins are typically on, and the genes are capable of synthesizing the differentiated product of the mammary gland, which is like lactoferrin and casein. Remember, we talked about casein a little bit before, and whey acidic protein. Or so remember, if you're a weightlifter or someone in fitness, you probably heard of casein and whey proteins. Those are from milk or cheese or whatever dairy product. So those are all off in the typical state. So when these cells are then moved from a plastic petri dish and placed on basal lamina, the genes for cell division proteins are turned off, so they don't divide anymore, and the genes encoding inhibitors of cell division, the genes that encode for inhibitors of cell division, such as P21, are on, and then the genes for lactoferrin are turned on. So they turn on genes lactoferrin and P21, which are genes for lactose production, while genes for cell division are off. So this is an early stage, right? So as the mammary gland wraps around, so we've turned off cell division, but we haven't turned on the mammary stuff yet, right? Besides lactoferrin. So we start forming these tight junctions around these mammary cells. And at this final stage here, once we get to the basal lamina completely surround, it forms this secretory or secretory epithelium, which allows for those proteins to be excreted, or secreted, I should say, right? So this is kind of, it waits until these tight junctions are formed and it's fully encapsulating the mammary cells, because you don't want to start producing and secreting. It doesn't have the mechanism to secrete yet, so you don't want to start producing all these milk proteins and producing milk before you're ready, right? So at the very beginning, we turn off cell division because you don't need to form a whole bunch more of these mammary cells. We start forming these tight junctions here, and so you start turning off some of these on some of these genes. So in this intermediate stage, we start producing casein. We've already started lactoferrin, and we stopped P21, stopped cell division, so that happened early, and we're stopping it. We still haven't turned on c-mic or the whey protein yet. But when it gets fully wrapped up, then we turn on the whey protein, and then we turn on the casein and the lactoferrin and everything to do its mammary function. And that is it. So that is an example of this interaction between mammary or the basal lamina or the extracellular matrices and cells, and how they this interaction can change gene expression and tissue specification, things like that. So thanks for watching, and I'll catch you in the next one.